For our project, we decided to talk about nucleic acid extractions in regards to point-of-care diagnostics. What is nucleic acid extraction? Nucleic acid extraction is one of the most pivotal steps in molecular biology. It is used in many areas of the biological and medical sciences since it is a starting point in any molecular diagnostic. I have included a diagram of this extraction, and we'll get into the steps in a few slides. So the history of nucleic acid extraction. The original method for this extraction was introduced in 1869 by Friedrich Miescher. He was studying the chemical nature of the nuclei of white blood cells, and he came across the very first form of nucleic acid extraction. In 1953, Rosalind Franklin, James Watson, and Francis Crick determined the structure of DNA. And this discovery granted them the Nobel Prize in Medicine in 1962. Their work, which every science person has studied, showed that DNA was made of two strands of long chains of identical nucleic acid or individual nucleic acid molecules known as nucleotides. These chains wrap around each other to form a double helix complex. DNA became known as the blueprint for cells and therefore all life forms. And I have included a DNA double helix here, which shows these individual nucleic acid molecules, the nucleotides on the phosphate backbone. In 1968, Gobind Coran, Marshall Nierberg, and Robert Hawley won the Nobel Prize for demonstrating how the sequence in which the nucleotides are joined together in strands of ribonucleic acid could be translated into chains of amino acids to make proteins. And this is one of the primary building blocks of cells. These discoveries have given a clear understanding of DNA and how nucleic acid extraction is used and why it's important. And then I've included a picture of a polypeptide chain, which also shows all the amino acids that it's made up of. So the 3 steps of nucleic acid extraction include cell division or lyse, which is breaking open the tissue and cells. The second step is removal of membrane lipids, proteins, and other nucleic acids. And the third step is purification and preservation of nucleic acid by transferring it to water or a buffer solution so that they will remain preserved without interfering with subsequent work. So when looking a little closer at the first step, which is cell disruption, also known as lyse, it is achieved by physical and or chemical methods whose main aim is to disrupt the cell wall and chemical membranes. Disruption methods are based on properties of the sample, meaning there is a wide range of tools and approaches that are used to achieve this disruption. Some methods include mechanical disruption, which relies on the use of rotating blades to grind and disperse large amounts of complex tissue. Another method is liquid homogenization, which involves the cellular tissue suspension through a narrow space, which shears the cell membranes. Another method is sonication, which uses pulses, high-frequency sound waves, to agitate the lyse cells or tissues. Another method is freeze-thaw, which involves freezing a cell suspension in a dry ice ethanol bath and then thawing the material at room temperature. This causes the cell to swell until it breaks. And the last method that I've included is manual grinding, in which a tissue is frozen in liquid nitrogen and then crushed using a mortar and pestle. And nucleic acid extraction methods. When looking closely, there are many forms of methods, and they can all be categorized into one of 2 different types, either a solution-based method or a solid phase-based method. Solution-based methods involve grinding frozen biological samples and then mixing them with solutions of chemicals devised to make it possible to purify RNA and DNA. Solid phase-based methods cause nucleic acids to bind to solid supports such as magnetic beads coated with silica and washed with alcohol to remove contaminants. The support is washed with liquid that renders the nucleic acid soluble again, which frees the DNA from the support in the process of elution. Some simple solid phase based methods. The simplest method for this is to add a solution containing a chitrope to the crude nucleic acid extraction. Chitopic agents are molecules that disrupt hydrogen bonding network between water molecules. In the presence of chitopes, nucleic acids are far less soluble and will bind to glass, silica-coated beads, and other solid supports. This makes it easy to separate them for contaminants.
Going off the other methods, one of them is cesium chloride gradient centrifugation with ethidium bromide. In this method, it is based on the phenomena of buoyant and specific density. Since cesium chloride is a salt, then when the solutes are subject to centrifugation at high speeds, then the cesium chloride sets up a concentration gradient. This is where it is super concentrated at one end and then less concentrated on the opposite end. In this case, where the centrifugation process is gently stopped, then the centrifuge will slow down slowly, which will cause the denser solution, in this case, the cesium chloride concentration, to settle at the bottom of the tube. The other molecules then separate themselves based on how dense they are. The denser substances will stay at the bottom, and the less dense ones will move to the top. In this case, the DNA is in the presence of a molecule called ethidium bromide, which will bind to it and become fluorescent when under the UV light. The DNA molecules also move towards the center of the tube when the ethidium bromide adjusts the density of DNA. The impure substances will then move to a different position, which leads them to be separated from the DNA, which can be easily seen and recovered since it'll be fluorescent under the UV light. Then the next step will be to separate the DNA from the cesium chloride and ethidium bromide. This technique is particularly effective for separating plasmids. And it is also easy to separate mitochondrial and plasmid DNA. These nucleic acids are arranged in supercoiled circular patterns. And since DNA is not supercoiled, it makes them easy to be separate from the plasmid DNA.
Another method for nucleic acid extraction is salt phenylchloroform. And an aqueous solution of salt allows for effective purification of RNA when mixed with the solvents of phenol and chloroform. The samples are first ground up and mixed with chloroform, phenol, sodium acetate, and salt. Then they are subjected to centrifugation, which causes the salt solution to separate from the solvents. This provides an upper aqueous phase and a lower organic phase that consists of the solvents. RNA will remain in the aqueous phase while proteins, other nucleic acids, and other contaminants will move to the organic phase.
And then the cetyl trimethylammonium bromide, NAE, this is mostly used for plant samples. And then the nucleic acids are insoluble solutions of 2% central cetyl trimethylammonium bromide nucleic acid extraction at high pH. Whereas the neutral polysaccharides and proteins are soluble. Because of this, it is easy to remove the plant-based contaminants.
Chilex extraction is utilized mostly in the forensic field for DNA extraction from different sources such as blood stain cards and hair. It uses resin that binds to common inhibitors of the polymerase chain reaction process. This yields a crude sample that preserves DNA and renders it useful for PCR-based forensic analysis.
The alkaline extraction is used for plasmid DNA isolation, crude preparations, or as the first step in all plasmid purification processes. This involves harvesting the bacteria of interest from a culture media and consecutively exposing the bacteria to a highly alkaline solution. The alkaline extract is mixed with the detergent sodium dodecyl sulfate, which removes proteins and contaminants. The advantage of this method is that the DNA is removed along with the protein, since there are many protein bounds to a large chromosomal DNA. And then a little bit of background that relates to biology 1 in our lab is this relates back to week 9 when we did a DNA extraction lab using strawberries. Since strawberries have a lot of DNA, which was surprising, and since strawberries do have a lot of DNA, they have 8 copies of each chromosome. They are considered octoploid. So there is a lot of DNA to isolate, as you can see in the image to the right. And I labeled where the DNA is. It's like the foamy white substance above the extraction buffer. And then understanding of nucleic acid extraction. When trying to understand the nucleic acid extraction of DNA, RNA, and protein, one has to know that this extraction is the basic method that has been used in molecular biology for many years. These molecules can be used to isolate from any biological material for preparative purposes, such as using nucleic acid extraction biology 1L to extract DNA from strawberry to analyze the pure DNA we get. The intracellular nucleic acids are known to be categorized as genomic plasmids and different types of RNAs. Nucleic acids show similar basic biochemical properties, even though RNA possesses uracil and DNA presents thymine. Nucleic acid extraction is divided into like 3-4 steps that were mentioned before. Cell disruption, removal of the membrane lipids and proteins and other nucleic acids, nucleic acid purification and binding from bulk, and lastly, nucleic acid and concentration.
So the critical thinking and analysis for our articles. So for our group, we believe that the article we chose, specifically the current nucleic acid extraction methods and their implications to point of care diagnostics, had organized information that went into depth. We thought the article was well reported because of the way it was organized and flowed. The paragraphs are straight to the point and delivered the information in a manner in which anyone can comprehend it. This article was what helped lay the foundation of our presentations. So the other articles we included in our references cited were good as well, but this was kind of like our go-to article, and it just served more of like a template. So now we're gonna go on to what is unexplained and unknown about nucleic acid, about the extractions. So there are many things that remain unexplained or unknown when it comes to these extractions. The scientists are still trying to figure out the easiest way to perform such complex extractions because within these processes, researchers encounter lots of obstacles. So as mentioned before, there are various extractions, and each one has their own complex steps. So we do encounter lots of obstacles. So as an example, a major obstruction for the development of an easy-to-use solution for the point of care process is the integration of sample preparation protocols into the portable devices. So removing these interference and extracting the target molecules are no trivial task, especially due to the vast differences among sample matrices as well as characteristics of the target analyte. So basically, the sample matrices consist of either blood or like a nasal swab or even saliva, and because they are so different, it can be kind of hard to limit them into just using them into one portable device. They each kind of have their own characteristics to them. So that is one obstruction. The second one is the microfluidic platform is a very cost-effective solution for sample preparation because of the reduced sample and the reagent size. So there we go. This is the microfluidic platform, and as you can see, it is very complex, but what they were talking about was that notice how in step D, there's just a little sample being used, so this can come off as very costly. And we also have to take into consideration that sometimes we don't even yield a lot of results. So yeah, and then this next slide include the various microfluidic devices. So as we can see, in diagram D, we see the nasal swab, and then the microchip in A, and it just kind of shows the different sample matrices and how different devices are being used. So continuing on, this next point I'm about to make refers to our tertiary source, which was a textbook we used, the Nucleic Acids Protocol Handbook. They brought up the issue that some tissues, especially skin, are very difficult to break up, and conventional methods yield too little and strongly degraded RNA. So getting into more depth about that, the biopsy material is often limited, and this biopsy material, so RNA becomes rapidly degraded in this biopsy material, where we cannot immediately conserve it during the extraction. So in order to conserve it, we have to break up the tissue entirely to get the RNA from the inner parts of the biopsy where the RNA degradation has not yet proceeded so far. So just going back to that bullet point up top, it can just be very difficult to break these tissues, knowing that RNA is very fragile, and we kind of have to be aware of that. So understanding the extractions from a researcher's point of view. So basically, some brief history about a researcher's understanding is that even though such extractions can give us a lot of obstacles, as I previously mentioned, researchers see these extractions more of as a way of learning how the extraction process works. So the main researcher's focus is how it actually works and how can we make this process easier. So for example, on the microfluidic platform, it is known for depending on external syringe pumps for fluid manipulation, but that limits applicability. So they developed several pump-free microfluidic methods such as centrifugal microfluidics, digital microfluidics, hand-operated microfluidic, and a capillary-based microfluidic device. So that just kind of shows how they are trying to figure out more convenient devices so that we can use it on a more well-rounded scale and be able to apply a lot of samples within it. So then our conclusion, why is this important and how does this relate to us? So in general, the use of these extractions are important and interesting because it can help cure diseases for all humans, plants, and animals. And if we target these sequences, we can figure out the mutations and oddities of the DNA which help develop the treatment for such diseases. So in simplest terms, this last paragraph is basically saying that extraction is all about detecting diseases and using this information to our advantage. It is our pathway to medicine, and it is how we are able to create these treatments. And then this is our references cited, and that is all.